Mi sentite adesso?
Sì, io ti sento.
Ok,
Sì, ok.
perfetto. Allora do il buongiorno, anzi la buonasera a tutti. Adesso in attesa, il professor Barouch, come già annunciato, arriverà alle ore 16. Quindi oltre a salutare tutti, vorrei salutare anche i rappresentanti delle istituzioni che hanno reso possibile questo seminario. Sì, ok. Perfetto. Scusate che ho un briciolo di ritorno. Ecco, perfetto così, scusate. Bello della diretta. Allora, dicevo, vorrei salutare i rappresentanti delle istituzioni, il professor Riccardo Zucchi, che è presidente della Scuola di Medicina dell'Università di Pisa, la dottoressa Daniela Corda, che è la direttrice del Dipartimento di Scienze Biomediche del Consiglio Nazionale delle Ricerche, e il dottor Gianluca De Bellis, che è il direttore dell'Istituto di Tecnologie Biomediche, istituto che è anche l'istituto della mia co-chair, anzi della chair di questa iniziativa, la dottoressa Elena Levantini. Li vedo tutti e tre collegati, quindi passerei un attimo la parola al professor Zucchi per poi, diciamo così, fare un brevissimo saluto, visti anche la presenza di numerosi studenti della nostra università e magari altri collegati via YouTube. Grazie.
Certamente, grazie a te Ranieri, sono felice e anche onorato di introdurre questo seminario a nome della Scuola di Medicina dell'Università di Pisa. E in questi giorni e in questo tempo tutti parliamo di Covid-19, di pandemia Covid-19. Una riflessione che mi è capitato di fare anche ieri l'altro, che facevo un corso di orientamento agli studenti, è che la classe medica e scientifica in generale in questo contesto sul campo si è comportata molto bene, con alcuni risultati quasi eccezionali, per cui sviluppare un vaccino in meno di un anno, e sono personalmente molto interessato a sentire il resoconto di uno dei leader di questo processo. Si è comportata forse un po' meno bene, a essere buoni mi verrebbero parole un pochino più pesanti dal punto di vista comunicativo, perché siamo stati bombardati in questo periodo da comunicazione, messaggi, suggerimenti di esperti o presunti esperti medici o scientifici, che purtroppo non sono stati del tutto consonanti fra loro, a volte hanno addirittura aumentato la confusione, con conseguenze anche al di fuori dell'ambito strettamente scientifico e medico, perché hanno sollevato speranze, paure e preoccupazioni non basate realmente sui dati, non basate realmente sulla evidenza scientifica. Per cui la riflessione che facevo appunto l'altro giorno e che torna a proposito oggi, che c'è veramente bisogno di sentire le opinioni di esperti di livello internazionale, di livello mondiale, che facciano un update di qual è la reale situazione, che costituisca la base per discussioni scientifiche e anche in un ambito più ampio per decisioni politiche, nonché per parlare con i pazienti, che purtroppo i pazienti stessi spesso sono molto confusi, specialmente quando si parla di vaccini. E questa è una cosa che succede oggi. Sono particolarmente felice che molti colleghi, molti studenti, in particolare moltissimi dottorandi hanno partecipato e sono presenti a questa iniziativa, perché sono sicuro che sarà l'occasione da un lato di apprendere qualche cosa e dall'altro di esercitare il loro spirito critico quando si tratterà di tradurre questo qualche cosa appunto in discussioni scientifiche, in discussioni che abbiano anche un'applicazione più ampia. Per cui mi congratulo certamente con te, Ranieri, con la dottoressa Levantini per aver saputo organizzare questa giornata. Ringrazio fin da ora il professor Barouch per la sua disponibilità. Auguro a tutti un buon lavoro e spero e auspico che questo sia anche un punto di partenza per un processo più ampio di coinvolgimento di un uditorio così variegato in temi di grande attualità, e nonché un altro punto importante che mi preme sottolineare, come questo sottolinei l'efficacia e l'interazione fra istituzioni diverse. Questo evento è formalmente e anche realmente organizzato congiuntamente all'Università di Pisa e dal CNR. Il punto di forza di Pisa, ma anche il sistema universitario italiano, è stato da tempo proprio l'interazione fra università e CNR, quindi sono particolarmente lieto di condividere la responsabilità e l'onore di organizzare questo evento. A questo proposito mi sembra molto opportuno cedere ora la parola a un insigne rappresentante del CNR, come anticipato anche il dottor Ranieri. Cederei la parola al professor Gianluca De Bellis, che è direttore dell'Istituto di Tecnologie Biomediche del CNR. Quindi grazie, buona giornata e bocca al lupo a tutti.
Sì, un attimo, è arrivato il professor Barouch, io lo inserisco all'interno della nostra stanza virtuale. Elena, se vuoi per cortesia istituzionale salutarlo, e poi diamo subito la parola.
Sì, direi senz'altro, direi forse senz'altro passiamo direttamente la parola al professor Barouch. Veramente 2 secondi per dire che mi fa molto piacere ciò che ha detto il professor Zucchi, cioè dell'interazione molto importante tra ICNR, che è un'istituzione nazionale, con le università distribuite su tutto il territorio nazionale, appunto. Questo è veramente un punto di forza. E ultimissimo, un ringraziamento ai 2 giovani ricercatori, Elena Levantini e Ranieri Bizzarri, perché è un'iniziativa che è partita dal basso, assolutamente meritoria, che ora siamo qui a goderci. Non do altri commenti e soprattutto non svolgo io l'attività per la nostra direttrice di dipartimento Daniela Corda, che mi ha detto: partecipo molto volentieri al seminario, fai tu l'introduzione alla cosa. Il professor Barouch è arrivato, quindi oltre a ringraziarlo direi che possiamo passare direttamente a Elena alla presentazione del suo collega.
Okay, so thank you everybody for your presentation. And then thank you for having accepted this invitation. So can you hear us, and can we hear you? Can we just make a little test? Okay, great.
Yes, hi, this is Dan Barouch joining. A pleasure to be here.
Yes, thank you so much. So I'm actually going to introduce you for 2 minutes, and thank you for taking the time. I know your schedule is extremely busy, understandably so in these days. So if you give me the green light that you're almost ready to share, I will give a 2 minutes introduction, and then we can get this started.
Sure, are we getting started now or at 10 o'clock?
If you want, we can start the introduction. I can start the introduction now, and then at 10 o'clock, we can start with your slides.
Sure.
Okay with you?
And I was planning to talk for about 30 minutes, and then I'm not sure the schedule of the day, but I think that was the instruction. So I was planning to talk for about 30 minutes, and then if there's time, I'm happy to take any questions.
Yes, definitely. That's great, actually, because as I mentioned, this is a combined event between the National Research Council of Italy and the University of Pisa, and we have a lot of students that are connected. So I think this is also a great opportunity for them to ask all the questions to a leader in the COVID-19 field. So basically, I can get this started, and I can say good morning, everybody, good afternoon and good evening. I know also people are connected from Asia, besides the United States and Europe. And so thank you, Dan, on behalf of myself and the institution, and Dr. Corda, De Bellis, Professor Zucchi, and Dr. Bizzarri from the National Research Council of Italy and the University of Pisa, for having taken the time, despite your busy schedule, to come here and tell us how only in 14 months you have been able to develop a single-shot vaccine, which is remarkable from a logistic point of view, given we have to vaccinate the entire world. So I'm just gonna say a few words on Dr. Barouch's CV. He's a scientist of stellar accomplishment, and he obtained his PhD in immunology at Oxford University, his MD at the Harvard Medical School. I'm going to read this information. He's professor of medicine and immunology at the Harvard Medical School, director of the Center for Virology and Vaccine Research at Beth Israel Deaconess Medical Center, and a member of the Ragon Institute of Massachusetts General Hospital, Massachusetts Institute of Technology, and Harvard. And also, he's part of the Bill and Melinda Gates Foundation collaboration for AIDS vaccine discovery. So his laboratory has long been focusing on developing novel vaccine and strategies for HIV-1 infection. And recently, he also used his expertise to tackle other diseases of global relevance, such as Zika, tuberculosis, and most recently, SARS-CoV-2. So we needed a vaccine to tackle the COVID-19 pandemic. And this is what he did in an unprecedented pace, thanks also to the many years that Dr. Barouch dedicated to basic research. And so the knowledge that he acquired in all these years of studies just came to fruition just in the right time. And so I personally feel, and I'm sure that this audience really shares my feeling, that it is very important to talk about this major scientific accomplishment in a moment in which the entire world is talking about vaccine immunology and research and vaccines. And so we need to mention that Rome was just not built in a day, and it takes a commitment of 24/7, 365 to deliver therapies. And so we need this major work from experts of the caliber of Dr. Barouch. So without further ado, as scientists, we are honored to have you here. As also your BIDMC colleague, I am proud to introduce this talk. And as human beings, we're really thankful to all you have done, because you are contributing to saving millions of lives. And of course, we wish we could have you here in Pisa, maybe mask-free, and maybe this may happen in the near future. But it's also important that we can deliver this information on this platform, because there are so many people that are streaming, and there is considerable value to that, given it would be for sure inspiring to witness firsthand the translational relevance of your research then. So then the floor is yours. And I'm actually going to turn my camera off. Should you need anything, we are here. Again, thank you for taking the time to give us this lecture.
Well, thank you very much, Elena, for those very kind words. And it's really a pleasure to be here. I would love to come visit you all in Pisa at some point in the future when conditions allow. But for now, I'm delighted to spend the next period of time with you. Let me share my screen. Host disabled participant screen sharing. So I think someone will have to allow me to share my screen. If that can be done, then I'm happy to present my slides.
Ranieri, are you going to take care of that? Ranieri?
Yes, I'm coming. Let me see.
Well, as I'm getting my slides loaded up, what I'd like to do in the next 30, 35 minutes or so is describe more of a personal journey of what we've done over the last year or 14 months in the development of one of several of the vaccines that are now available to the United States and to the world for COVID-19. It's by no means the only one. I will touch on the broader picture of the field as well, but I'll talk mostly about what we've done that has resulted in the J&J single-shot vaccine. But in no way am I saying that this is the only vaccine or the best vaccine. What we need is we need all the vaccines, and all the vaccines that are licensed are very safe and very effective. Okay, I think I can share my screen now.
Okay, here we go. Thanks.
Okay, can you see my first slide?
Yes, we can. Yes.
Very good. Okay, so what I'll tell you is really more of a personal journey. So our journey started on January 10th of last year. That was actually the day that we were holding our annual Barouch Lab retreat, coincidentally. And we were reviewing work done over the past year and plans for the future. And we did review the fact that there was a cluster of mysterious pneumonias in Wuhan that had spread to 41 cases and 1 person had died. We found those numbers to be alarming. The fact that an unknown pneumonia could spread to 41 individuals and was lethal enough to cause at least 1 person to die made us think that we might need a vaccine for this illness. But of course, you know what's happened in the last year. In just over a year, instead of 41 cases, we now have 126 million cases. And instead of 1 death, we have over 2.7 million deaths. It's truly astounding how a viral pandemic can rip through our planet and our society and affect each and every one of us in such a personal and profound way. However, the science and medical and vaccine communities have stepped up in a way never seen before in human history. Over 200 vaccine candidates have been developed according to the WHO, a truly astounding response from vaccinologists who saw very quickly that a vaccine was likely going to be the only solution to this pandemic. Essentially, all research platforms for vaccines have been deployed against SARS-CoV-2, including DNA vaccines, RNA vaccines, proteins, vectors, inactivated viruses, and more. I would argue that the most important timeline is actually not the timeline to show clinical efficacy, which has been achieved by maybe a half dozen vaccines so far. What is most important is the timeline to produce and to deploy billions of doses of vaccine worldwide to stop the pandemic. In the United States, we have 3 vaccines approved for emergency use. The first 2 were the mRNA vaccines from Pfizer and Moderna, and the third one was the Ad26 vector vaccine from J&J. And in the United States, we might soon have the AstraZeneca vaccine and possibly the Novavax vaccine as well. I'm of course aware there are a number of additional vaccines produced from Russia, from China, and elsewhere that are also very, very important for worldwide vaccination. But the COVID vaccine field was essentially, had a giant leap forward on November 9th when Pfizer released the initial results of their phase 3 study, showing for the first time stunning efficacy against this illness. Just a week later, Moderna announced the results of their phase 3 study, also showing a very high efficacy rate in their phase 3 study. And later on in November, AstraZeneca released the results, not of their US study, which was just released a few weeks ago, but of their studies in the UK and Brazil. And then in the end of January, J&J released their top-line efficacy numbers of the Ad26 based vaccine. So in the United States, there are 5 vaccine programs that have reached the point of phase 3 efficacy trials. These are the Moderna and Pfizer mRNA vaccines that showed efficacy in November and were approved in the US in December. The J&J Ad26 vaccine, which I'll talk more about, the launch was in September and the efficacy was announced at the end of January and was approved at the end of February. And then the other 2 vaccines that are being studied in the United States are the AstraZeneca vaccine with the University of Oxford, the ChAdOx1 vector that started phase 3 trials in August, and the efficacy results for the US study was announced about 2 weeks ago. And finally, the Novavax adjuvanted protein vaccine. The study started in December, and we hope to hear efficacy results in April, possibly May. So how did the global community step up like this? How is it possible to develop not one, but multiple COVID-19 vaccines in just about a year? Vaccine development for a new pathogen usually takes many years, if not decades. So the development of a COVID-19 vaccine, or rather multiple COVID-19 vaccines in just a year, is truly unprecedented. Some people have been concerned as to whether vaccine development has been rushed. In fact, nothing has been rushed. Everything has been done as rigorously or even more rigorously as for any prior vaccine trial. And every step of the development of every one of these vaccines has been under the eye of public and scientific and medical and regulatory scrutiny. So I would say that these vaccines have not been rushed, but they were developed very efficiently. And so how is that possible? Well, actually, these vaccines were not developed in a year. The platforms that these vaccines rest on have been developed over decades. So the fundamental reason of how vaccines were developed so fast is they represent the culmination of decades of advances by hundreds of researchers in virology and immunology, as well as in particular, the development of novel vaccine platforms that are essentially plug and play, gene-based vaccines in which you can just put in a cassette of a new antigen. And that technology, which is the underlying technology for the mRNA and the vector-based vaccines, is really the core reason why these could be developed so fast. These vaccines used vaccine platform technologies also that were tested for other pathogens. So they could be advanced into clinical trials very quickly. And development activities were performed in parallel rather than in series. For this to happen, there has to be substantial tolerance of programmatic and financial risk. But most importantly, we cannot compromise any patient safety or regulatory integrity, because ultimately a vaccine that's not trusted and not used is going to fail in its mission. So one example is that virtually all of the large companies started to mass produce millions of doses of their vaccine while the clinical trials were still ongoing. Clearly, that's a financial and programmatic risk, because if the clinical trials fail, then millions of doses would literally just be thrown in the trash. But nevertheless, it was felt to be worthwhile for COVID-19, so that if there is a positive result of the efficacy trials, and if there is approval, then there can be millions of doses distributed essentially that same day. Also, for COVID-19 vaccines, mass production and global distribution is required, because the goal is to achieve herd immunity on a global scale to end the pandemic. We can't just distribute vaccines to subgroups of individuals or individual countries. There really has to be a global approach, because not until the entire world is vaccinated can any of us be safe. So our work on the Ad26 vaccine platform didn't start in 2020. It actually started in 2003, so 18 years ago. So to some extent, the development of the J&J COVID-19 vaccine, to some extent, started about 18 years ago. And one of the first projects that my lab did when I started my laboratory was to develop a series of new vaccine vector technologies. Not for coronaviruses, but actually for HIV, because HIV vaccines was one of the major focuses of my lab from when it started, really up until today. So we developed a series of adenovirus vectors, about a half dozen adenovirus vectors, including Ad26. And this is actually the first report of the initial cloning, vectorization, and selection of ad vectors that we decided to move forward. We first moved Ad26 forward, again, not for coronavirus, but rather for our HIV vaccine program. And over the last 15 or 17 years, we've developed a candidate HIV vaccine based on Ad26 vectors. And interestingly, this vaccine will likely read out in efficacy trials later this year as well. But HIV, of course, is a very high-risk virus, because it has certain immunologic and virologic characteristics that are unprecedented in vaccinology. But nevertheless, the development of Ad26 initially for an HIV vaccine candidate is really the cornerstone of why this vaccine technology was developed and then became available for other vaccines. Our first use of Ad26 as a potential pandemic response was in 2016, when we developed, together with our industry partner at J&J, an Ad26 Zika vaccine. And we wanted to see how the vaccine platform would perform if we used it for a so-called well-behaved virus, as opposed to HIV, for which there are many scientific challenges. So we used Ad26 for Zika, and we were really quite impressed that a single shot of the vaccine could protect monkeys against Zika challenge. And here in a clinical trial in 100 individuals using 2 different doses, a low dose and a high dose, either a single shot vaccine or 2 shots of the vaccine, then we showed that a single shot of the Ad26 vaccine, shown in the blue, the light blue and the yellow, was sufficient to induce antibody responses, neutralizing antibody responses, in essentially 100% of individuals. And these responses lasted for more than a year. Now, if we did give a boost on day 57 to 2 of the groups, here shown in the orange and blue triangles, we were able to boost these responses considerably. So what we learned from the Ad26 Zika program is that we could develop this quickly in a pandemic response, in a real-life pandemic response setting, and that a single shot of this vaccine was sufficient to induce neutralizing antibody responses in virtually all recipients. And this formed the initial concept of the single shot vaccine, this study for Zika. So if we fast forward now to January of 2020, we decided to develop Ad26 as a COVID vaccine together with our industry partner at J&J. Because J&J was our partner for HIV and Zika, and they had the platforms to develop Ad26 at massive scale. So we decided to develop the replication incompetent common cold virus called Ad26 for COVID-19, really because it was in place and had shown promise for other pathogens. I told you about HIV and Zika. J&J also developed an Ad26 Ebola and RSV vaccine. Prior to the pandemic, the vaccine has shown to be safe and immunogenic in over 100,000 individuals. There was also regulatory approval by J&J for the Ad26 Ebola vaccine in Europe. And J&J showed that mass production and global distribution was feasible for Ad26 Ebola. Also, as part of the Ebola program, because it had to be distributed to Central Africa, J&J invested in formulations and developed a formulation for Ad26 vaccines that did not require sub-zero freezing, which obviously would be difficult for the developing world. And so that's actually where the lack of sub-zero freezing requirements originated. There was also pediatric experience with Ad26 RSV to babies down to 4 to 6 months of age. So we reasoned that it could be developed as a pediatric vaccine quite quickly also. So we thought that fundamentally this would be useful to explore for COVID-19 as a single shot vaccine with no frozen cold chain required, which would conform to the WHO target product profile for our COVID-19 vaccine. So this is what we did. On January 10th of last year, we started this program. The virus sequence was released by the Chinese researchers, and we basically started working on the sequence that same day. That was a Friday night. And we gathered 4 of the scientists in our group to start poring over the sequences and doing alignments and developing electronic sequence immunogens. When we got back to the lab on Monday, January 13th, we were ready to start ordering synthetic genes and designing the vaccine. By the end of January, we had an agreement signed with J&J to develop an Ad26 COVID vaccine. In February, we started preclinical studies in mice and rhesus macaques. Also, by the end of February, we had a virus challenge stock to do preclinical studies. And in early March, we started to develop small and large animal models of disease. We really settled on hamsters as small animals and macaques as large animal models. At the end of May, we had our first publications demonstrating natural and vaccine immunity in non-human primates, not specifically for this vaccine, but I think important for learning about immunology and vaccinology in general. By the end of July, we started our phase 1/2 clinical trials, and also we published the protective efficacy of the Ad26 vaccine in non-human primates. And these really led to the September 21st initiation of the single shot phase 3 clinical trial. About 2 months later, we also started another 2-shot version of the vaccine, and that study is still ongoing. And then, as you probably know, at the end of January, J&J announced interim safety and efficacy data from the single-shot phase 3 study. At the end of February, FDA granted emergency use authorization. And during this calendar year, J&J is committed, and so far fulfilling that commitment, to produce and deploy at least a billion doses of this vaccine for worldwide use. So I'll go through a few slides from our July paper, which was the study that we showed that a single shot Ad26 vaccine protected against SARS-CoV-2 in rhesus macaques. Again, this was inspired by the Zika data, and this study essentially led to the clinical or supported the clinical program of a single dose vaccine. So we tested not 1, but actually 7 different versions of this, the spike protein in Ad26 vectors in collaboration with our Janssen partners. I'll just focus on the bottom one, what we called at that point in time the SPP vaccine, because this is the vaccine that ultimately did the best and went into clinical trials. It has a native, a wild-type leader sequence. It's a full-length spike with furin cleavage site mutation and the diproline stabilizing mutations that many of the vaccines incorporate. The protection data was really quite impressive. We had a large sham control group, and these are monkeys that were challenged by the intranasal and intratracheal routes with SARS-CoV-2. And these are viral loads in bronchoalveolar lavage following challenge. You can see that 20 out of 20 sham control monkeys were infected and had high levels of replicating virus on day 2. And these were generally controlled by about day 10. In the SPP vaccinated animals, there was complete protection in bronchoalveolar lavage. The other 6 vaccines also showed relatively good protection with 0 to 2 breakthroughs for each group. In nasal swabs, it's typically harder to protect in the upper airways than the lower airways. The sham controls, all of the sham controls had virus replication in the upper respiratory tract, and this was generally controlled by about day 10 to 14. In the SPP vaccinated animals, 5 out of 6 animals were completely protected. 1 animal had a low blip of virus. The other 6 groups also showed a moderate or a modest to moderate degree of protection in nasal swabs with 1 to 3 breakthroughs per group. And so this is the summary of protective efficacy in this study. These are peak viral loads following challenge in bronchoalveolar lavage and in nasal swabs. You can see the sham control group had about 5 logs of virus in BAL and 5.5 logs of virus in nasal swabs. And in the SPP vaccinated animals, there was no detectable virus in BAL and almost complete protection in nasal swabs. Because of the large size and the variety of outcomes in the study, we were able to do an immune correlates analysis, and we showed that the neutralizing antibody titer induced by the vaccine, either 2 weeks or 4 weeks after immunization, inversely correlated with peak viral loads in BAL. And thus, these data strongly suggested that at least if we were not concerned about variants, then the neutralizing antibody titer likely correlates with protection. So this study showed that a single shot of the Ad26 vaccine could protect macaques against SARS-CoV-2 in the lung as well as in the upper respiratory tract. We reasoned that a single shot vaccine, particularly one that didn't have sub-zero freezing requirements, would have important advantages over 2-shot vaccines for mass immunization and pandemic control. But we also knew that a second shot of the vaccine would typically increase neutralizing titers by a substantial amount, a half log or a log. So we thought that clinically it would be important to test both a single shot vaccine and a 2-shot vaccine. We also showed that neutralizing titers were the strongest correlate of protective efficacy, consistent with the prior DNA vaccine study that we did. And so the development plans for this vaccine, which was renamed for clinical development, Ad26-CoV2S, was to start phase 1/2 clinical trials on July 22nd. Which was in 1,000 individuals, led by J&J. Our site at BIDMC was participating. And then the first phase 3 efficacy study was initiated on September 24th. So September 21st, essentially with the week 4 immunogenicity data from the phase 1/2 study. So you can see how tight the timelines were. This was a 45,000 person study led by J&J with the NIH-sponsored CoVPN network. So the initial results of the phase 1/2 study were published in these 2 papers, one in the New England Journal of Medicine, one in JAMA, about the immunogenicity in the phase 1/2 studies. And in short, the vaccine was safe and immunogenic, similarly immunogenic in humans as it was in animal models. So cohort 1 was young adults, 18 to 55, and cohort 3 was elderly adults over 65. And there were 5 groups in this study that received in a randomized blinded way, either placebo, a single shot of a low dose of the vaccine, 2 low doses of the vaccine, a single shot of what we call high dose, which was really just 2-fold more, 10 to the 11th viral particles, and then 2 shots of the higher dose of the vaccine. And what we can see here is that the placebos were mostly negative. At baseline, most individuals were negative. The few that were positive, we think those were probably individuals who were infected during the spring surge of last year. But what we can see very clearly is that by day 29, almost all individuals had a positive antibody response here by ELISA. And if you look at the summary curve on the right, then the groups that received a single shot vaccine continue to go up slowly up through day 71. So there's no drop. There's actually a slight increase from day 29 to day 71. And the individuals that were boosted on day 57 had about a 3-fold increase in antibody titers. Similar antibody titers were observed in the elderly individuals. If we look at neutralizing antibodies, we see a very similar trend in that almost all individuals develop neutralizing antibody titers by day 29. The single shot vaccine led to an increase of neutralizing antibody titers through day 71. But individuals that were boosted on day 57 had about a 3-fold increase in neutralizing titers. And similar neutralizing titers were seen in elderly individuals. Also, what we reported, and now we think is of even more relevance, is the T-cell responses. This vaccine induced robust CD4 as well as CD8 T-cell responses in the majority of individuals within 2 weeks following vaccination. So COVE 3001 was the phase 3 randomized double-blind placebo-controlled trial of the efficacy and safety of a single shot of this vaccine. This was fully enrolled as of December of about 44,000 individuals. There was a 1-to-1 vaccine placebo randomization. This was a single shot of the 5 × 10⁻⁻ viral particle dose. And this was a truly global study with 3 main geographic areas: in the United States, in Latin America, including Brazil, and in South Africa. Now, something very frightening and important happened during the course of this trial, which is that during the course of this trial, there was the emergence of new SARS-CoV-2 variants that theoretically could threaten vaccine efficacy. So the pandemic, as I'm sure you all are aware, is much more complex today than it was several months ago. And now studies from many labs have reported on these variants, and in general show that the variants that include the E484K mutation leads to some degree, in most cases a substantial degree, of escape from neutralizing antibodies, both natural immunity as well as vaccine immunity. And the impact of this clinically is seen because of the reinfection rates in South Africa as well as Brazil with the variants. So the variants largely fall into 2 different categories. Some, like the B117, first reported in the UK, lead to enhanced transmission and possibly increased mortality. Others, such as the B1351 variant first reported from South Africa, as well as the P1 and P2 variants first reported from Brazil, might also have an even more concerning property in that they can partially evade neutralizing antibodies. And therefore, the concern is whether these might lead to reduced vaccine efficacy. And indeed, I think that's what was seen with a number of vaccines tested in South Africa. So the J&J phase 3 trial really converged from a timing perspective on 2 important events. One was the emergence of these variants. And here I'm just showing the variants in South Africa. And the efficacy collection period of December, January really precisely overlapped with when the B1351 variant essentially exploded in South Africa. So essentially in South Africa, the J&J vaccine was really tested essentially explicitly against this variant. Over 95% of circulating viruses or sequence viruses in cases were the B1351 variant. In addition, the December-January efficacy collection period coincided with the peak global surge in the US, in Brazil, and South Africa. So it's a very complicated epidemiology in that the force of infection was dramatically higher than last fall. Most people probably were multiply challenged, multiply exposed, as well as variants emerging and circulating in South Africa with over 95% of cases, in Brazil with over 70% of cases, and even in the United States with the California variant and others. So despite this very complex epidemiology, the J&J study proceeded in multiple countries at the peak surge of the global pandemic, including in South Africa with 95% B1351 resistant variant. And the protection against any symptomatic infection was still fairly good, with 72% in the US, 68% in Latin America, and 64% in South Africa. So only a very modest, interestingly, a very modest reduction of protection despite the P1 and P2 variants in Brazil and the B1351 variant in South Africa. But even more important than protection against any infection was the protection against severe disease, which was 85% globally. In fact, it was 86% in the US, 88% in Brazil, and 82% in South Africa. So really no decline, so robust protection against severe disease, and really no decline of severe disease protection despite these variants. And then there was also 100% protection against hospitalization and death. So against the most important outcomes of severe disease, hospitalization, and death, this vaccine actually shows robust and equivalent efficacy against the P1, P2 variants in Brazil and the B1.351 variant in South Africa. So these are just some data from the actual clinical trials showing protection against severe COVID disease. There is some protection as early as day 14 and up to 85% protection by day 28. But that's actually not even the end, because these are the Kaplan-Meier curves of protection against severe disease, they really separate on day 7. So there is demonstrable protection that starts even 7 days after a single shot vaccine, which is quite astonishing in my view. And the endpoints of the trial that were predetermined, which are of course then the ones reported, are the day 14 and the day 28. But I think you can see that these curves continue to separate, and that goes along with the increasing immune responses. The antibody and T cell responses continue to increase through day 70. And in the trial after day 48, there were actually 0 cases of severe disease in the vaccine group compared to the placebo that continued to accrue cases. And this really led to the FDA that authorized use in the United States of this vaccine on February 27th. And then there was rapid additional regulatory approvals over the last month in Canada, in the EU, and the WHO, and I believe others as well. So I'll also very briefly mention that there's a second phase 3 trial that's ongoing now, which is called COVE3009. The target enrollment is 30,000. I believe it's finished enrolling, 1-to-1 vaccine placebo randomization. And this is testing a 2-shot version of the vaccine. Essentially the same vaccine, but given twice instead of once. And now that the studies are unblinded and the placebos are getting the vaccine, this trial is actually going to morph into a head-to-head comparison of 1 versus 2 doses, because the placebos will be, right now the placebos in this trial are being called back to get the approved EUA vaccine, which is a single shot vaccine. So interestingly, this study still has a lot of rigor, and this study will then be a head-to-head comparison of the single shot vaccine versus a 2-shot version of this vaccine to determine whether or not a 2-shot version gives better efficacy than a single shot. And this is also a very international study in the United States, in Europe, in South Africa, and in Asia. So where are we with the summary and the rollout of this vaccine? Safety and efficacy has been demonstrated in a global phase 3 study, including in the US, Latin America, and South Africa. There's high efficacy against severe disease in all regions studied, in all subgroups, including against the resistant variants that we're hearing so much about. We believe a single shot vaccine with no sub-zero freezing are beneficial and may facilitate mass vaccination campaigns, both in our country, in the US, and globally. And J&J has a commitment to supply the world with at least 1 billion doses of vaccine this year. And with a partnership between J&J and Merck, it might actually exceed that. The emergency use authorization has been approved by the FDA in the US and is rolling out in the US right now. I believe this week, 11 million doses are being rolled out. So over the last month, probably 20 million Americans will be vaccinated with this vaccine. And it's also approved in Canada, the EU, WHO. I believe I'm hearing news reports that this vaccine will start to be deployed in Europe starting next month. And there's also an expanded access program deploying this vaccine in healthcare workers in South Africa as they await their own approvals. So I'll close with perspectives. Vaccine development for COVID-19 has proceeded faster than for any pathogen in history. I don't think these vaccines have been rushed. I think they've been developed very rigorously, but in a very efficient way. And of course, safety and public trust are critical. We strongly believe that multiple vaccines need to be implemented in parallel, since we need to accelerate the vaccine rollout in the US and throughout the world. Viral variants require further study, the vaccines may need to be updated and additional variants will likely emerge. In fact, one of the strongest reasons to accelerate the pace of the vaccine rollout throughout the world is not only to protect the individuals and their families and loved ones and society, but also to drastically reduce the amount of circulating virus to help prevent the emergence of new variants in the future that might be even more problematic than the ones we have today. And finally, we have to realize we are a global community of 7 billion people, and we need multiple vaccines to be deployed. In fact, all vaccines that have proven to be safe and effective, I think, need to be deployed as quickly as possible in the world to help end the pandemic. I'll just close with this slide. I'm often asked, when will the pandemic end? And of course, I can't answer that any better than any of you can. But I do think that in the future, the pandemic will end. And in the future, when historians write about everything that happened with the COVID-19 pandemic, there will be a whole lot of things that went horrible and things that went horribly wrong. But I think that the shining light will be seen as science, and the ability of science to step up, the ability of science to develop vaccines as well as treatments and public health measures, but particularly for vaccines in a relevant period of time to help end the pandemic, I think will show the world the power and the importance of science to do good for the planet. I'll stop there. Thank a lot of people in my lab, as well as at J&J and many other groups. This is a slide for the NHP studies. I apologize, I don't have a slide for the clinical program, but this is a picture of our lab at Beth Israel Deaconess Medical Center. This picture was taken about 1.5 years ago in pre-pandemic times when we could gather like this as a group. And we haven't had a picture like this since then, but hopefully with vaccination, we'll be able to get together like this again before too long. Thank you. I'll take any questions if there's time.
So it's a terrific job. So congratulations for what you have said, for all that you have done. And it would be great to have fellows around and be participating to this wonderful seminar, but unfortunately we are here, so we might start with some questions. If there are some students that they can type away the questions on the chat. And but I will ask Dr. Daniela Corda if she's got a question for Dr. Barouch. Otherwise, I will start myself.
First, yes, here I am. Wait. Okay, here I am.
Okay.
No, I followed that was very interesting. And while you were talking, I was thinking that we had recently, a meeting with Rino Rappuoli that I'm sure you know from GSK. And it's interesting how you stressed that, all the process of vaccine, making this vaccine was absolutely not rushed through. This is a very important message because people, especially after the AstraZeneca problem that were in Europe, few people had problem after AstraZeneca. There is this idea that a vaccine can be dangerous, which is totally wrong. So we should really develop a better information. And someone before said that this too, that we need really better information. So I was wondering, apart from the great work you did, and I don't have any specific question on that, I wonder how the information is going in the United States. Do you have many people that, are kind of cautious toward or actually enemies of the vaccine, or are you better than us in this?
Yeah, thank you for that question. So, as doctors and scientists, we often think that the data is the only important thing, and that a lot of the public perceptions are not very important. That is simply not true for vaccination, because the public perceptions are absolutely critical. because if enough people are hesitant to get the vaccines offered to them for one reason or another, real or imagined, then if we don't achieve herd immunity, then we will not be able to end the pandemic. So unlike many areas of science and medicine, public perception is actually critical for the overall goals. And so I think it's very, very important for doctors and scientists, as well as governments and stakeholders and companies, to be very clear and careful of how they communicate with the public. There has to be openness and transparency and honesty first and foremost. But there also has to be a balance. I mean, not all news is good news, and everything has to be balanced in a way that doesn't skew public perceptions in an incorrect or inappropriate way. And absolutely, if a vaccine is not safe, then it should not be used. But if a vaccine is safe and effective, then it absolutely should be used. So in the United States, I think we've seen a major shift of public opinion. Vaccine hesitancy in the United States reached a fever pitch, was incredibly high last fall, say in September, October. I think that was really fueled by two major things. First, we did not yet have data on safety and efficacy of any vaccine in large numbers of people. So doctors and scientists and vaccine developers, we could not actually tell people for sure these vaccines were safe and effective because the trials hadn't been finished yet. Secondly, the development of these vaccines, particularly last fall, became intertwined in American electoral politics. And because of how divisive that is here in our country, then that led to all sorts of negative perceptions. And without getting to the specifics, then anytime a medical issue is politicized, then many people have very strong feelings, largely emotional, one way or another. And that can never be good for medical intervention that you want to be accepted by everybody. So vaccine hesitancy in the US reached a peak that we've never seen before last fall, ironically, as we were starting a surge of a disease that was killing people. So it was completely counterintuitive, but anyway, that's what happened. So what we saw over the course of the time period from November to January, we saw a substantial decline of vaccine hesitancy. In my view, largely because those two things no longer existed. So the election was over, so it was no longer linked with electoral politics. And secondly, we did have data from not one, but multiple vaccines that they really were safe and highly effective in large groups of people. Then from January to March, we have actually seen a further decline of vaccine hesitancy. I was just reading an article last night that the number of Americans who appear to, or who report saying that they will either probably or definitely not get the vaccine has gone from 22% down to 17% between January and March. And it was somewhere around 40% last fall. So it's really declined from about 40% to 22% in January to 17% in March. And that's actually largely driven by increasing feelings of safety and acceptance among African Americans in the South, who obviously have historical reasons to distrust medical care. So vaccine acceptance is drastically improving in the United States compared with where it was last fall. And my own personal view, or hope rather, is that as long as things continue to go very smoothly, which they have gone remarkably smoothly in the United States, that number will continue to decline. And if it can decline from 17 down to maybe the 10% range, there obviously will be some people who just will refuse, and there's nothing you can do about that. But I think that of the remaining people, of the remaining 17% who are still hesitant in the United States, I actually think that many of them will actually ultimately want to get the vaccine once they see that it's safe in hundreds of millions of people, not just millions, but hundreds of millions of people showing its safety. And and high levels of efficacy, proven efficacy, not just in clinical trials, but on population bases. And basically, when they see their own friends, their own family members, their own neighbors being vaccinated, and then being able to engage in many other activities and getting their lives back to normal, I would imagine that many of the people who remain hesitant will want to get vaccinated. I am aware that in Europe, I think the vaccine hesitancy in certain countries in Europe is higher than the United States. And hopefully in all countries, vaccine hesitancy will start to decline as the evidence supporting safety and efficacy of the vaccines essentially is skyrocketing.
Thank you so much for elaborating on this aspect that might seem a minor aspect, but is a major aspect because the information is not always as accurate as it should be. For good thing and for bad thing. I mean, and in the specific case of vaccine, since you are talking to a school of medicine, it's important that our young student will be, our ambassador in disseminating science and good information. So thank you very much for this question.
Let me just add one other comment, since I guess I can't see all the students, but if I can speak maybe directly to the students and young doctors and young scientists just for a minute. You folks are not only the future of science and medicine, but I think that scientists and doctors need to take a more active role in communicating with the public. And if you speak the truth, you speak honestly, people will listen because of your training, your expertise, your stature in society. And we need more doctors and scientists communicating with the public, and rather than leave it to other groups of individuals who might not have your training and expertise. So I would encourage all of you not just to make the contributions to science and medicine that I know you will, but also to be the spokespeople to the general public about what needs to be done from a scientific and medical perspective.
Thank you.
So there are a few questions actually, and I would like to mix one of those with one of mine. That is basically what determines the pattern single shot versus two shots? I mean, is that formulation of the platform, or is so this is something that many people are asking now, for instance, in Italy, because we are going to have the vaccine from Johnson & Johnson, and there will be a change. So people ask why we should start with 2 doses with other vaccines instead. Of course, there are scientific reasons, but is there a major platform that will determine this, or it's just a matter of how you select the antigen? I mean,
Well, to some extent, it's based on the platform, and to some extent, it's based on how they were studied. So the vaccines can only be approved based on the clinical trials that were done. So if, for example, if the J&J study was, if the first study was done as a 2-shot study, it would be approved as a 2-shot vaccine. But the reason why we did it as a single-shot study is because the platform had proven to be effective in Zika as a single-shot vaccine, and then a single-shot vaccine was sufficient to protect monkeys, and then in the phase 1 trials, a single-shot vaccine was sufficient to raise the immune responses that we thought would be protective. Also, unlike some modalities where a single shot may raise immune responses, but they tend to be transient, for the Ad26 platform, a single shot vaccine gives immune responses that continue to go up over time, so that even 10 weeks later, the immune responses are still rising. So there's not a need to protect against with some modalities, the vaccine responses either are not very visible after a single shot, or if they are, they wane quickly. And for reasons we don't completely understand, the durability of the responses with Ad26 are really quite good and continue to go up for months before they sort of stabilize. And so that's one of the reasons why we thought that the I-26 might be useful as a single shot vaccine. And for a pandemic setting, then it has obvious logistic benefits.
Can I step in and just ask a question then? I mean, you were mentioning this rising, and then do they get to the plateau? How long do they stay at the plateau, and how long, if you have the chance to notice that in your models, when do they start going down, if they do? And how much fast do they decline?
Well, for the COVID-19 vaccine, only time will tell, because we don't have data at the next time point yet. We will. So we have data through essentially 10 weeks. Which is about almost 3 months, but we don't yet have the 6-month data yet, because it just hasn't been enough time.
I was wondering about the models, not about the clinical trial, but if you had any models, like animal models that were used, if you could observe anything there. Maybe you're not there yet. Probably.
Yeah, so we have 6-month follow-up time points from monkeys, which show stabilization, but really no decline in responses. And we have 12-month data from the Zika data that I showed you that shows fairly good stability of responses even a year later. So I think only time will tell. There could be antigen-specific differences, but what we know is that the responses continue to go up for at least 3 months. And then our suspicion is they will probably approximately stabilize for a period of time.
Okay, then I have another question, and then I'm going to leave the word to other people. Do you think that people that were exposed to the virus, and so they have been asymptomatic, can get it rather safely? And also people that got it in a symptomatic way, do you think they could be exposed to this vaccine? What is your thought about it?
Yeah, the advice is that everyone should be vaccinated regardless of whether you've been infected before or not. Exactly. It turns out if you were infected before, then you have some residual antibodies, usually not very high, but you probably have some antibodies that might give you partial protection, but probably not as good protection as you get with a vaccine. So everyone is recommended to get the vaccine, even if you've been infected before. But if you've been infected before, actually, then you'll probably have some of the highest responses to the vaccine. You'll probably have even better response to the vaccine than someone who hasn't been infected by before. And in fact, in the United States, there's a debate going on for the 2-shot vaccines, such as Pfizer and Moderna. Then do you actually only need 1 shot instead of both? And there's actually an immunologic reason why that makes sense, but there's no efficacy data yet with giving a single shot of the RNA vaccines to people who were previously infected. For the J&J vaccine, it doesn't matter because it's only a single shot vaccine. So for the J&J vaccine, then people who have been previously infected would still be recommended to get the single shot vaccine.
I was asking again on the view of what we were saying before, right, that I hope that the right knowledge is going to become viral. I'm just going to use this pen, becoming viral, so that people listening from you are an expert, also our student, understand exactly how to respond to questions from the general public, because this is a major question that is going around, and hearing from you, it's really very important for them. So that's why I was reiterating the question. But thank you again.
Sure.
There are a couple of questions from the students. The question is, the security in children is is under testing right now. I think so. And what is supposed to change? And there's some change in the formulation, the kind of vaccine, just a matter of trial.
So I know that Pfizer, Moderna, and J&J I know have pediatric programs. Actually, just this morning, Pfizer announced that they finished their cohort of 12 to 18, and their vaccine looks safe and immunogenic in that cohort. So Pfizer is a little bit ahead, but Pfizer, Moderna, and J&J are all planning to dose down to I believe 6-month-old babies over the next few months. I believe AstraZeneca is doing the same thing. I'm not as familiar with their plans, but all of the main vaccine developers have pediatric programs, and the goal is that we need children vaccinated. Certainly, school-age children we need vaccinated ideally before they go back to school in the fall. Whether we'll achieve that or not is not clear, but that would be the goal in my view. The main reason why they can't just be added on clinically now is because we don't know the dose to give. We don't know whether for any of the platforms, RNA or Ad26 or ChAdOx1, we don't know whether a child 12 years old or 2 years old or 6 months old should get the adult dose of the vaccine, or whether they should get maybe a lower dose. And so the reactogenicity and immunogenicity needs to be tested in children, often at a couple different doses. So the vaccine will be the same, but the question is, will the dose be the same as adults, or will the dose be different? And if the dose is different, then at what age cutoff?
Okay.
So that's the reason why children can't just get the vaccine now.
Another question is, so this is something that popped up also in the public opinion sometimes. Why were the RNA vaccines to be the first to be developed, and because they are supposed to be a very novel technology? Well, not that novel if you consider that many experiments have been done 10 years ago at the starting for this technology, but nonetheless, why were those vaccines were on the front line?
Well, RNA has a lot of advantages, and one of the main advantages is that it can be manufactured very quickly, because you can manufacture RNA synthetically. You don't need to grow it in cells the way you do with either vectors or, say, inactivated viruses or proteins. So RNA can be made essentially synthetically, essentially in a beaker rather than in cell lines. And that means that they can be made more quickly. And so that's one of the reasons why the RNA vaccines were sort of the first out of the block.
Okay. Another question is, in the scenario where basically everybody needs to be vaccinated every year, is there the need to change something? I mean, it's supposed to be a different approach in terms of, I mean, how to realize a vaccine, or how it's supposed to be formulated or not? Or you just go straight this way? I mean,
Well, I think it remains to be determined whether people will need an annual booster shot or not. My hope is we don't. And my hope really rests on the fact that, at least for the J&J vaccine, there is proven efficacy against the variants. So at least for the variants that exist today, then we know that the J&J vaccine has high efficacy. Our presumption is that the other vaccines also would have high efficacy, but that's not yet proven. So if that is true, then we may not need annual booster shots. However, if new variants emerge that more fully and more completely escape from vaccine antibodies, then there's a possibility that we'll always be in sort of a cat and mouse chase, like we are for influenza, where new variants emerge and then vaccine developers have to remake vaccines every year, and everyone has to get, a booster shot in September before the winter surge. That would be very difficult to implement on a global scale. I think, certain countries, maybe in the US and the EU, could do that. But given how hard it is to vaccinate the world, it would be really, really hard to vaccinate the world every single year, I think. So I think time will tell, and it really will be the population impact studies on whether we need to do that or not.
Should we rely a little bit on some convergent evolution? So, because I read on Nature a couple of days ago that there are some reports on convergent evolution almost anywhere in the world about COVID-19. I mean, the SARS-CoV-2. So maybe it's not that variable as you might like.
So, I mean, my lab is, before COVID-19, we were primarily an HIV research lab. And for HIV, there really is essentially limitless diversity. Whereas for SARS-CoV-2, it looks like there is not going to be limitless diversity. We think that the virus still has other pathways it can evolve into. So I don't think we've seen everything yet. In fact, I think that there's a good chance there will be new variants that we really haven't seen. But there does also appear to be convergent evolution. And in many regions of the world, say the 501 mutation crops up to better adapt to the human host. The 484 mutation comes up as one of the principal mutations to evade antibodies. But we're not seeing the same sort of limitless diversity that we see for HIV. And so that gives us hope that if we can, if it's a more of a finite problem rather than an infinite problem, then we might be able to have a set of vaccine constructs that can vaccinate the world and can anticipate essentially all future variants.
Any other questions? No questions from...
May I ask you one, let's say, political question? Which has been the role of the public hand, let's say, of the government in supporting your effort in getting to the final positive result you showed us?
Well, so I'll divide your question in two parts. First is our work over the last 17 years on HIV. And that is largely NIH-sponsored research grants. And so the NIH has played a very important role in supporting our work over the last 2 decades that developed the underlying technologies for this vaccine. Now, the specific development of the I-26 COVID vaccine had multiple sources of funding. Some of our very initial early funding was really philanthropic funding through BIDMC, through the Reagan Institute, and through other sources to allow us to start working on something immediately. But the bulk of the funding for the I-26 COVID-19 vaccine is really the United States government through BARDA, as well as J&J through their internal funding. So essentially, sort of half and half. The initial development, it's public knowledge. The initial development, I think, cost about $1 billion, and about half of it came from the U.S. government, and half of it was paid for by the company.
Okay. So I think
if I have time, just a very quick biological question, I would say. How did you choose your preclinical models? And what about the spontaneous course of the infection in these non-human models, either mice or monkeys?
So we actually looked at many different models. We looked at multiple mouse strains. We looked at hamsters. We looked at ferrets. We've looked at cynomolgus macaques. We looked at rhesus macaques. I'm also part of the International WHO Working Group on Animal Models. So starting in February, then essentially virtually all of the labs in the world, throughout the world, Russia, China, Europe, US, South America, all converged on weekly phone calls led by the WHO on the development of animal models. So we were all really seeing all of the animal models being developed in real time. And so with our own data, then we observed that the virus replicates well in either ACE2 transgenic mice or in hamsters, less so in, a little bit less so in ferrets. And since so we settled on the hamster as a small animal model. And it replicates fairly similarly in multiple different macaque species, including cynomolgus macaques, rhesus macaques, African green monkeys. And so we really use cynos and rhesus macaques interchangeably for a large animal model.
Okay, so it's about 5 p.m. here in Italy. So we probably, I don't know if Dan can stay. I got some more questions from another auditorium because we are online with YouTube. So but I don't want to be too much. Can you stay a little bit longer?
I have another commitment that's starting now, but maybe one more quick question. Would that work?
Okay, so I'll take. What about the interference between the anti-flu vaccine the classical anti-flu vaccine and vaccine for COVID-19? Is there any?
Well, they're very different vaccines. The flu vaccine is a whole killed virus, and generally has to emerge every year with a hope for a match. Here, the nature of viral evolution for SARS-CoV-2 is very different than the nature of evolution for influenza, and the platforms are different. So they're really completely different vaccines.
Okay, Dan, thank you so much. Thank you so much. I think we really enjoyed conversation with you. I'm sure we also have members of the press. If they have questions for the Italian audience, they can also get back to us. And if there is anything major, we can ask you and then we can relate to them. And I really hope that everybody, all the people that participated in this meeting are going around and spreading the news that science, serious science can help. And we need to trust science to help us all. So Dan, thank you so much for having taken the time and go back to your amazing work. Thank you so much.
Thank you all. Thank you all.
Thank you.
I appreciate it.
Bye.
Bye.
Thank you very much.
Thank you.
Allora, approfittiamo per salutare tutti. Grazie.
Esatto.
È stato un seminario estremamente informativo, molto importante. Sono molto contento che ci sia stata la possibilità, il tempo e la disponibilità di Dan Barouch. Volevo ringraziare in particolare Elena per la sua connessione diretta con Bettisa Deaconess e naturalmente tutti coloro che hanno partecipato anche solo ascoltando. Salutandovi, vi auguro una buona sera a tutti.
Benissimo, buonasera.
Grazie a voi.
Grazie a tutti, arrivederci.